Okay, so let's actually see what this looks like. So I told you there's a lot of data in there. Let's actually do a short tour so that you can see this and you can follow along yourself. I mentioned I was going to do visual system, but we've had so much input on this male-female liver data set that I thought maybe I'll just show it to you. So if you go to Gene Network, let's see how many of you can get on without a denial of service. But you got it? Yeah, if you don't get it, let me know. I kind of am curious to see what kind of load we can handle. So here are the genotypes. Let's look at the genotypes, just so that you can see what dumb old genotypes look like. There was a question about how you encode genotypes. So let's say I wanted to look at a genotype that's located on chromosome 1 between 10 megabases and 12, well, let's make it 20 megabases. So that's the search term. There's no reason for you to remember these search terms, but basically you can see here's sort of the idiot's guide to the search terms. They're like baby SQL queries, but they have a simple structure. If you learn these, you're a power user. If you spend 5 minutes to learn these, you're like one in 500 people in terms of your expertise. So it's easy to become an expert. So let's find, so we're just saying, give me a genotype that's located somewhere on chromosome 1 between 10 and 20 megabases. It says it's a confidential database. I don't know why it's confidential. It certainly shouldn't be since it's 2005. So let me just reload that. Should be, okay, genotype. So I retrieved a total of 5 markers. They have the RS format, reference SNP, RS is what that stands for. They're all in my criterion, and I could do the same kind of analysis that Steve mentioned. Let's say I decided that this SNP at 14 megabases was a really cool SNP. I could just pull it down, and I'm just showing you the dumbest sort of phenotype. So a genotype is essentially a phenotype. It's a molecular phenotype, right? So in the good old days, before we had genotypes, we used visible markers, coat color. Tail, you had a kinky tail. Not that anybody here has, but for mice, they would use that. And then this is what the genotypes are encoded in Gene Network. So they're just ones and zeros, basically. There will be some minus ones. There's zeros. Those are heterozygotes. So we coded around 0, where 0 is a heterozygous value. And there should be twice as many heterozygotes as either of the homozygotes. So let's go look at that. So the mean value of the genotypes is 0. That's what we expect. The minimum is -1. We could look at that as a probability plot. Here it is as a probability plot. Kind of boring, and expected z-score. Here it is as a bar chart where the zeros don't show up. But this is the most boring but kind of useful type of phenotype, a genotype as a phenotype. Now I can get more interesting. I can say, what is the correlation between this particular marker and gene expression in the liver? So the way this is structured in Gene Network, you'll see we've got our univariate statistics. Basically, there are 3 bands here: univariate, bivariate, and then gene mapping tools. And you'll want to do multivariate, or complex analysis. So basically what you do is you then export things into your shopping cart. There's a shopping cart metaphor here. See where that add symbol is? I can put that in my shopping cart. I could put ApoE. I could put every gene associated with autism in my shopping cart. So now I've put that in my shopping cart. And now I can say, hey, tell me what genes in liver covary. So here are the tissue types I have: adipose tissue, brain, liver. And I'm going to do it across, we've put the same data has gone into Gene Network 3 ways. This is a little bit dumb, but it's the way we do it. There's a male data set, a female data set, just like the data sets that you guys downloaded for the demo. And then there's a joint data set, all of them. So here's liver, males and females together. And I can say, please just compute the Pearson product moment correlation. It's not really, it's actually a biserial correlation because the way, well, maybe not quite. But I'm going to compute the correlation here. And now Gene Network does everything it can do on the fly, it does on the fly. So we try not to pre-compute. We don't want to cram you into a particular model. We want you to be able to build your own models. So let me show you why. Let's say that Jake Luce has called me and said, Rob, you know, we shifted our plate and actually all of these turned out to be -1s. So I can go in here and I can actually change these to -1s. And, you know, if you've got a paper and it's almost in press at cell and one reviewer is nagging you that you don't have enough significance, you just go in and tweak these parameters, right? Just joking. That's not really why we do it.
You're on video.
We really do it so you can find the outliers and take care of them. So I was telling a story at lunch. I spent $10,000 of my wife's hard-earned money to apply for a patent that was due to an outlier. Had I looked at the data more carefully, it would have never happened. And so it's usually there so that you can detect outliers and fix them. But now I could recompute the whole correlation with my messed up data, or I could just say, please reset it. So there's the reset button. But now these are the correlations. Let me make this a little bigger so you can see these. So these are the correlations between a single SNP and phenotypes. Now these are kind of unusual phenotypes because they're mRNA expression data, but it could be triglyceride levels in serum, right? It tells us what the gene is, what its ID is, and these are the same IDs and numbers that you'll see in your downloaded. Here are the correlations. So I can say there's a high correlation between that particular marker and this particular gene or transcript. I should call it transcript. I notice that in this course, we always talk about genes, but actually we're doing very little with genes. We're dealing with mRNAs. So if you do start writing grant applications and papers, keep it clear, because as soon as you start doing this kind of expression genetic analysis, very difficult for the reader to know whether you're really talking about the gene or the transcript. So we're talking about transcripts here. And if I click on this transcript, it'll actually show me the correlation coefficient. It'll be kind of an odd-looking correlation because one of my traits only comes in 3 states. So we have the 3 genotype states. This is BB, this is BH, and this is HH. In this case, it's not the BXDs that I told you about. This is a black 6 crossed to a C3H. And the C3H is carrying an ApoE mutation. And you can see there's a group difference between the 3 genotypes. And that's what I meant when I said, I had my hand model, right? There are the 3 genotype states, and here's my phenotype, and obviously there's quite a bit of covariance between those. So this is what mapping looks like. And we just have to do this systematically. So the implication here is that this SNP likes this mRNA. So let's look at the mRNA. So here I'm going to click on the mRNA for transmembrane protein 70. I have no idea what it is. Here's the expression data. Some comments about the expression data that we're using in this tutorial. Somebody asked a question, what are these values? And Peter answered the question that they're logarithm of the ratio. Unfortunately, in this case, they're essentially z-scores, or they're z-score-like, so you don't know whether a gene is highly expressed or not. Everything has a mean of 0. I hate this scale because you don't know if it's got high or low expression. So what they should have done, and I wish Jake would do this, is basically could just add the constant back to the z-score. You'd still have your beautiful z-score and you'd know whether it had high or low expression, but I guess he can't get the data from Eric or Merck or whatever. So I have no idea whether this is abundantly expressed or not, but what I can do is now I can show you the third function here, is I can map it. And mapping is what I just showed you. I just showed you mapping for one marker and one mRNA. But now I'm going to do it across the whole genome, starting at chromosome 1, position 1, going to chromosome X, the last possible position on chromosome X. And I'm just going to plot my statistics, the p-values, or the likelihood ratio statistics, which is a chi-square value, just like Steve showed you, or the logarithm of the odds ratio, which is basically just minus log of the p-value. So all of these numbers are really simple. If it's a log score, just count the number of zeros after the decimal point. So if somebody says, I need a log score of 10 to the minus 8 to publish my paper in Nature Genetics, that means they need 7 zeros and a 1, right, after the decimal point. So let's do this. So what it's doing now, also again in real time, is it's taking that vector of data. So the data is about 400 gene expression data sets, half male, half female for this TMEM70. And it's doing the analysis and computing the p-values. If there's a big gap between two markers, so we got a marker here at 10 megabases and another one at 20 megabases, it says, whoa, that's a big jump. And it'll impute the state of the genotype in between and give us an estimated p-value. And this is what it looks like. So this is what we would call a whopper. And expression quantitative trait loci are often whoppers. So chromosome 1, chromosome 2, these are the permutation p-values down here. So you have to reach that pink line to be judged significant genome-wide. So we're way, way, way above genome-wide. If you divide that value of 196 by 5, you have the approximate LOD score. The direct conversion, the actual correction factor is 4.6. So we have, and this curious little symbol here, I don't know if you can see it, there's a small triangle right there. That's the location of TMEM7. So we're saying something very obvious but very important. TMEM7 regulates its own expression. So why is that so important? Because that now is a potential causal node. So you can say, not only do I know where this gene is located, but I know that there's a gene variant somewhere in the vicinity of that gene that modulates the expression of that gene. And then you can go and you can look at the sequence state and say, not only does that gene have variable expression that depends upon the haplotype that it inherited, whether it inherited the B or the H, but there also might be a missense mutation in it. So you can have two categories of variants. Basically, all the differences among us in this room are caused by two very simple processes. Differences that affect protein structure and differences that affect mRNA type that is transcribed. Transcription or translation differences, right? That's it. So this is half the picture. This tells us this gene has different expression levels, and we could just look in a sequence database and figure out whether there are differences in protein structure. So that gives you a really simple summary of the kind of univariate analysis. I haven't shown you anything too fancy, univariate and bivariate. But let me give you a flavor of where this is, and then I'll jump back into my presentation. I've got, these are the genes that covariate with my SNP, right? I have 500 of them in there. I'm going to select them all, and I could do a go analysis. So if I hit this go analysis here, it's going to export it to Web Gestalt at Vanderbilt. I could put it in my shopping cart. You'll see the add symbol right over here. You see I'm trying to wiggle right there. That'll move all 500 into my shopping cart. And now my shopping cart will have the SNP at the top, and then it'll have all of these mRNAs in there. And I asked my programmer to put all of the phenotypes in there. So later today, all of the phenotypes that are in your WGCNA work case, remember we have those 4 or 5 files. We have the male, the female, then we have the clinical, I think it's called clinical comma-separated values. All of those will be in Gene Network later today, and you'll be able to actually map the triglyceride levels, the body weight, et cetera, in the same data set that you've used for the WGCNA. So now, they look the same, but now this is my shopping cart, and I can export it. I could do kind of fun things. that you guys know how to do better than I do now using WGCNA. Obviously, this might be inappropriate for a WGCNA analysis, but we sure as heck know we should see a strong signal on chromosome 1, right? So if we had the biggest module we expect here is a chromosome 1 covariance controller module. And that would be essentially the eigengene of this should look just like our marker. And let's do a little bit of that. So I'm going to select just the first 10 of these. And you'll notice something about, let me just take you through this table. You guys in the back probably can't read this. Let me try to make this a little bit bigger so you can see it. Okay. So it says what the data set is, what the probe set number is, whether we know what it is. So here's Stoffen 2. Here's something that nobody figured out what it was, which is amazing. Because they designed the probe, they put it on the array, they spent big bucks putting it there, and they still have no idea what it is. So you'd say, why is that the case? And I have no idea. These vendors, they make their arrays and they don't bother to annotate them. It's amazing. So it maps, this actual aligns to chromosome 1 at 21 megabases. So if you wanted to find out what it actually is, you could just blat it and find out. Let me show you what else is there. Then we do database some summary mapping data. So you'll notice that this is where the gene is located. This is where the mRNA made by that gene maps to. So that's kind of bizarre. It's simple. So the gene makes the mRNA. We've got the mRNA sloshing around in the liver. We measure it as a trait, and we ask, what's controlling the expression level? And in most cases here, well, in every case, it's chromosome 1. Doesn't have to be. So let's find one that isn't chromosome 1. It's good. I mean, there's no problem here. Here's one that claims to be mapping to chromosome X. So that means that would be a transcontroller. Let's say there's some important transcription factor on chromosome X that controls the expression of a gene on chromosome 1. This would be an example. Are there any other? So you can see that basically we've got a chromosome 1 cluster. And I would caution you, I find that a lot of really smart bioinformaticists use data sets like this, but they're really not aware of this forced association that's due to linkage. So linkage is a pain in the ass. It happens in mouse populations. It's not quite so serious in human populations. The ideal situation would be every gene was on its own chromosome. That would be perfect. Then we wouldn't have linkage to worry about, right? They'd all associate and segregate randomly, and we wouldn't have to worry about haplotypes other than at the chromosome level. In a human GWAS, we're very close to that level. So if you do a human GWAS with 10,000 subjects, the haplotype blocks are so small that neighboring genes often have very small R-squareds. So that's really good, and that's why human GWAS is cool. The thing that makes human GWAS difficult is that you then end up doing 20,000 tests, right? Every gene now is a separate entity. Every time you do a GWAS scan, you're studying 20,000. So GWAS studies have tremendous precision, no power. And your question about epistasis, nobody does epistasis in GWAS because they just don't have the power yet, unless you have a specific model. So if you're looking for nonlinear interactions between gene A and gene B and they're on different chromosomes, you need sample sizes in the millions to really do that with any power. Mouse studies, on the other hand, have the opposite problem. They've got great power, but they have lousy precision. And so there's almost always a trade-off between power and precision. And people are always trying to find the perfect solution for both of these. There is no perfect solution other than to do two types of studies in one. And that's what I do now. I use human GWAS studies to validate mouse QTLs. It sounds backwards, but really it's not a dumb thing to do. And at the same time that you're validating your mouse QTL, you're making sure it's translationally interesting. Okay. Let me break now and just ask I know I had too much coffee before I started. That's occupational hazard. But are there any questions so far about this? Yeah.
So, did the people who designed or who analyzed the microarray data, they check that there's none of the SNPs are located within the probe? In other words, that the QTL, apparent QTL, is just an effect of the microarray differentially binding to
Yeah. Yeah, so this is a big problem in both microarray and RNA-seq data. We thought it wasn't going to be a problem with RNA-seq, but it turns out it's just a series of problems. The microarray is designed with sequence from C57 black 6 in the case of mouse. And if we do our RNA-seq alignment, it's also being aligned against C57 black 6 genome. Whoops, did I do something? Or? I'm still alive, okay. So you can mask, you can find these SNPs now because it's easy to sequence a mouse and figure out whether there's a SNP that overlaps your probe. Yeah.
I know because Jason Moore, he's doing the epistasis and he's building it. I know because I listened to his lectures.
I'll have to have a look at that, yeah.
So that's my
Okay. So the comment was there is a resource for doing epistasis in human populations using GWAS data? Okay, yeah, yeah. Usually if you can constrain your model, if you have some justification, you think this might interact with that, then you don't have the same multiple test issue. If you do a naive epistasis scan across the entire genome, you've got a million by million matrix, which probably boils down to about a 20,000 by 20,000 matrix, and the multiple test hit you take is huge. Thank you. So back to Peter's question about artifacts. So because you're aligning against reference genome and because what you're aligning to reference genome may not be reference at all, you'll end up with hybridization that's poor for one genotype and good for the other genotype or haplotype. And what you'll do is you'll see an asymmetry of effect direction. And so in Gene Network, we keep track of the effect direction over here, the minuses and the pluses, and you can look and see whether across the whole data set it's balanced or not. And so if you then took the mean of this vector across all 25,000 probe sets, and you saw that it was clearly like 5 standard deviations beyond expectation in the minus direction, you'd know that that was because you had black 6 bias in alignment. You see the same thing for RNA-seq. You can then say, okay, I've got this bias, I'm going to apply clever bioinformatic method X, I'm going to mask my SNPs, I'm going to align to virtual genome from the other strain. You can make virtual genotypes or virtual genomes, and you can correct that, and just make sure you have a nice symmetrical distribution around zero effect. Yeah, it's a big problem for human. So how many of you know about the GTEx effort? The genotype tissue expression, humongous effort. It's probably $50 to $100 million effort. Like everything else, it's at the Broad. Not that no sour grapes there. So they're taking tissues from 1,000 subjects and they're doing RNA-seq for 50 plus tissues, 10 plus brain regions, depth of read of 20 million reads per tissue. And there it's going to be a big problem, again, because you don't know, every individual is unique. So when you do the alignment, it's going to be difficult. You won't copy number variants, SNPs, probably the common SNPs you could mask for and kind of figure that out. But there's some serious issues in these data sets. You can check them out. Okay, any other questions before I kind of go back?
Yes, it might be a little bit off topic, but how did you manage to, or how did you convince your wife to give you $10,000?
I said it was my wife's $10,000 because I hear about it from her. I think only 5,000. 5,000 was mine, 5,000 was hers. I sort of think of it as high risk. In this case, it was high stupidity, high risk. I should have been on the lookout for the outlier. I'd be happy to show you guys the analysis that I did and how I made the mistake, or how, blame it on your colleague, how my colleague made the mistake. That's the nuts. So, oh, I remember I did the gene Go analysis. I took those 500 genes. Let me just show you this while I'm here. So this is the enrichment of the top 500 covariates of TMEM70. There's no reason why they should be enriched, but and you can't see this very well, but they are enriched for nucleus by chance, probably. Anyway, so the idea is now to, once we have these data sets, you can do kind of package analysis. You can go to Go. But the cool thing is, if you structure it right, you should be able to export it and then put it in and do a GCNA analysis. Typically, you'll probably want to export everything. So let's assume that you had so much fun dealing with the male and female liver data set that Steve gave you, which is this one, right? And let's say you wanted to check for the how much agreement you had between networks. Well, here's another UCLA data set. It's also liver, it's also ML, and it's also male-female. You could download that. And that's the one that I was looking at this morning. I was trying to figure out why it didn't line up with the data set you guys are using or were using, and the answer is I picked the wrong one. But Jake Lucis even has a third data set. So down here is a Castaneous F2 intercross and another liver data set, and also male-female. So right now there are 3 data sets for liver on the same platform, from the same lab, done over about a 2-year period, all run at Merck, and it'd be a great data set to see whether you have robust network conservation across these populations. The Castaneous it's like almost like a gorilla-human intercross. They're very different. So it'd be interesting to see whether that network is preserved. And in each case, you can download these. So let me just show you how you download it. So you'd say, I want this male Castaneous liver data set. You click on the info button. And then over here, it says, it doesn't let you download it. Sorry. I don't know why it doesn't let you download it, but normally it would let you download it. Let me just make sure that what? Okay, yeah, most of them will let you download it. Let me show you one where it would. So this is the BXD family. And if I want to download this data set, I click on the info button again. And in general, you should see a link like this one right here. And that allows you to download it as a zipped text file, usually in different formats. If there's a file you want and we haven't put it on there, let me know. We do not, I'm too lazy to put everything in Geo. So GeneNetwork is its own accessioning service. We don't put everything in Geo just because nobody's given me money to do it, although it shouldn't be that big a deal. If anybody wants to volunteer to put all our data in Geo, just contact me. But then we put the metadata here in many cases for the individual data set. So again, pretty easy to download all these data sets if you wanted to work with WGCNA at the level, which reminds me, I've got a data set for, where's Dan? Dan, are you in here? Oh, he stepped out, okay. Okay, I've got a 1 gig exon array data set for him to try his cool his splicing code on. Okay, let's go back and just, time is, we've almost got an hour into this. Let me go a few more minutes. Sure, let's have a 5-minute break, yeah.
Let's make it 10 minutes.
Sure. I don't know if that's... All right, is everybody refreshed and ready to do some practicals? So let's I'm going to take you through just a few slides, and then we'll immediately get back into a practical, and I'll ask you to do an analysis. So if you didn't bring a computer, just pair up with somebody or hang out, or If you want, I can walk you through it. But let's do a systems genetics analysis of the visual system. Or if you want, you can, again, choose your own adventure. If you're interested in Alzheimer's disease or Parkinson's, you can pick a particular gene or a network of genes that interests you. Let me tell you how to choose your own adventure. So I'm going to take you through this visual system example, but if you don't want to do that, let me just give you an idea. So I mentioned that you can automatically be a power user if you spend 5 minutes and read this. So let's say I wanted to find every paper that Steve Horvath has where his name is attached to a gene. I would put in name equals Horvath S. You have a middle initial, Steve? P. Yeah. People without middle initials, big problem. But you can do a search for a Go term. You can say, I want everything involved in axonal transport, and you could find the Go term, and theoretically it should work. If it doesn't, send me a little hate mail, and we'll try to get it working. The most fun one for many of you will be the RIF. So let's say I'm interested in autism. For some reason, I want to study autism in mice because I'm perversely convinced that mice are a good model for autism. And I want to study prefrontal cortex. And I actually believe that mice have a prefrontal cortex. I did my postdoc at Yale, where Pat Goldman-Rakish was one of the senior PIs, and I should know better than to think that a mouse has the nerve to have a prefrontal cortex, but I swear they do. So mouse prefrontal cortex, and then I'm interested in a saline control data set. That's what this is. And then I'm just interested in all genes that have been tagged with the word autism. So that's RIF equals autism. And what this is doing, RIF stands for reference into function. I don't even have to put the parentheses there. It will, every night we update the database and we say, has somebody claimed that this gene is associated with autism? And if they did us the favor of entering a reference into function into NCBI, we'll then pull it out. And this should give us a list of 600 or so, 800 now, 800 genes that have supposedly been tagged with the word autism. And I could let's just look at the very first one to see what's there and why it got tagged with the word autism. For all we know, it says ACE is most definitely never been associated with autism at all. So it could be a negative finding. So we want to know that. So this is what ACE looks like, and we go to this gene wiki, which is where all the RIF data hang out. And we can add our own data there too, by the way. But these are some tools, and let me just see what it says about ACE. So I click on the gene wiki tool. And again, you have trouble reading that. I'm just going to find the word autism. Whoops, what did I just do? I have to spell it right. High prevalence and severity of autism traits in RAS opathies compared. So there's one occurrence of the word, and that's the only occurrence for angiotensin converting enzyme. So that's really not a very strong link. So there are probably better ways to do this, but if you have your own favorite set of autism genes, you could certainly put them in there. So going back here, you could say, what if I just do a search for autism in a mouse, BXD, and let's say now I decide I want to go to look at a hippocampus data set. I'm going to make that my default. You can just see what I just did there, made it my default. And now these are 20 genes that have the word autism in them. And I could say, okay, do I see anything interesting here to pursue? So you can build your own networks this way, and I think probably 80% of you will want to work up from genes, but maybe, well, I don't know. Many of you will want to work down from phenotypes, neurodegeneration or learning and memory, whatever. So let me get back. But Again, just have a look. If you just hit the word gene network, it'll take you back to the basic search screen. Look at these searches and put one in and have some fun. Choose your own adventure. But here's the adventure that I chose. When I didn't start as a geneticist. I started as a physiologist and then a developmental neurobiologist. And I did a lot of cell counting. And the cells that I counted were retinal ganglion cells and cells in visual area 1 and lateral geniculate nucleus. And at some point I got sick of counting cells and I thought I would just measure mRNAs because it was so much easier. And it actually is easier, which is kind of odd. We have quite a bit of data on the visual system. And so if I do that, if you do this search in gene network for the BXD family, which is the family for which we have the best data, you will get lots of hits. And I hope you can read that. Let me again see if I can, nope, can't make that bigger. But this is retinal ganglion cell residuals. So that's just correcting for brain weight and some other extraneous variables that we think are nuisance variables. Sex, we actually think is a nuisance variable in this condition. Doesn't explain any of the variance, so we can just collapse across it. You don't have to do that, but eye weight, lens weight, lateral geniculate neuron number, glial number, and some other, here's ocular dominant shift, some electrophysiology, genetics, electrophysiology and genetics for ocular dominance shift, cortical plasticity. So that gives you a feel for the kinds of data. You can go to Gene Network. You've already seen this kind of data for a genotype. I don't think I ever showed it to you for a phenotype, but there's retinal ganglion cell number, and the blue arrow points to the mother, the red arrow points to the father for retinal ganglion cell. For photoreceptor number, and those are millions of cells, again, the blue arrow is the mother, and the red arrow is the father, and then here's eye weight. And those are nice univariate statistics, and now you want to say, let me put the whole visual system together and ask some questions. Do retinal ganglion cell numbers co-vary with photoreceptor numbers or with eye weight? Kind of very simple question, but for the most part, you can't answer these very simple questions simply because the data is not structured correctly. Somebody may have measured, even in human, may have measured eye weight in 1,000 human subjects or done tomography or imaging, and they may have counted retinal ganglion cell number in a separate population. But again, these are little isolated data sets, and there's no way to mash them together effectively. In the mouse population, we can mash them together, and we have error terms, which are really nice. So, and we can make the error terms as small as possible, or small as we can afford, simply by resampling. So the data sets that Jake Lucis did here, these F2 intercrosses, those mice are gone forever, right? You can't come back to them. Any one of these mice, you can come back to it. You can say, I want BXD49. I want to buy 10 of them from the Jackson Laboratory, and if you have $135 per animal, you can have them. I mean, it's pretty expensive. If you buy them from me and Lou, discount for this class, only $50. That's why I sell them to Jake Lucis, so he's been using these now. So, more data sets. So this is just sort of working up the visual system. So this is just showing you how you can generate these very difficult data sets, and they're not going anywhere. They just get better with age. Lateral geniculate neuron number, age-adjusted brain weight. Okay, so here's your task. I want you now to map a gene for brain weight in the BXD family. And I want you to tell me what gene you think is responsible for this variance. So that says 300 milligrams. And that particular case over there, BXD5, has a brain weight of about 520 milligrams. You can see we've got beautiful error terms here. Lulu and I went crazy on this study. It turns out it's relatively easy for us to measure brain weight. So in some cases, we have 100 animals per strain that we've done. So it'll give you a nice QTL. Okay, so go to Gene Network and map it. And when you know what chromosome it's on, let me know. And then we'll see whether we can actually clone the gene, build a network around it, find which candidate is most plausible candidate.
But I think you need to show us the commands how to map.
Okay, all right.
What is the green
All right.
You mentioned the red and the blue, but I'm seeing the green one.
The green one is the F1 hybrid. Yeah, so you can see there's quite a bit of dominance there. Okay, so Steve says I can't use the Socratic method. Okay, all right, so no Socrates. we're in California. Yeah, okay, all right, so you can watch me do it if you don't have it here. Okay, so I'm back to gene network. You can see that pretty well. Let me make it one step bigger. I'm interested in phenotype, brain weight. So I have to go to genotypes, phenotypes at the very top. So go to gene network. You want your page to look like this roughly. And again, there's lots of human data, there's rat data, there's Drosophila data, and for those of you who are into barley, and I don't know what else we've got in there, there's some other crazy species in there. The only one that we really work hard to keep it kind of clean is the mouse and the human, but we basically don't have enough money to actually do the software development and the data entry and the data cleaning. It's a lot of fun to do it, but there are only 5 of us working on this. Okay, so we've got that, and then we do a search for brain weight, and there are a couple ways you could do this, but the way I want you to do it, if you just put in the word Hager, so this is from a Nature Communication paper published 3 years ago. Reinmar Hager was one of the authors. Here are the 13 traits in this paper. They include different parts of the brain. So the top one I have is basolateral amygdala, brain weight, brain weight, brain weight, brain weight. So we have a bunch of different versions of brain weight, and they've been corrected for different ways. So we talked about removing nuisance factors, and so here we have a nuisance factor in how we measured the brain weight. Was it fixed or not fixed? So we now have a correction factor. How old was the animal? Was it a male or female? So we correct for all of those factors that we don't think are interesting. By the way, in mice, there's absolutely no brain weight difference between males and females, despite the fact that the males are a lot bigger. There's huge sample sizes. But we correct for it anyway because it doesn't really hurt. So I'm going to take this trait, 12661. So if you wanted to, you could just go to Gene Network and enter the number 12661. How many of you are sort of doing this live? 1, 2, 3. I just want to see. Let me know if it gets really slow, because I'd like to know if you're just not getting response out of the machine. I'm just kind of curious how much slow? Okay, if it's slow, that's interesting. Okay, so now here's a classic phenotype. It's different than a gene, so it doesn't have a lot of those cool tools, so there's no gene wiki, there's none of the links to ABA, et cetera. But it does have a really good description of phenotype, and I want you to look at this because I spend a ridiculous amount of time just describing the phenotypes. It's like, I'm like Mr. Librarian for this. So central nervous system morphology, and then there's a colon, and so that's sort of our controlled vocabulary. So you can look at everything that's central nervous system. Then we have brain weight, repeat measurements prior to, adjusted for variation, for dissection error, and then the reference. And then we can just scroll down, and we see all of our data. And in this case, it shows us the mean, the standard error of the mean, and the sample size that we had for that particular strain. And it automatically flags outliers. So if you're going to do a statistics web service for molecular biologists, we love molecular biologists, but they basically aren't very good at statistics, so you have to make sure that they don't screw up. So you want to flag the outliers for them. How many of you are molecular biologists? How many people did I insult simultaneously? 1, 2, 3. We love you very much. I wish I were a molecular biologist, actually. It'd make my life a lot easier. And I wish I were a statistician. That would make my life even easier. So we can sort this. So I just clicked on that, and you can see the top 2 are really outliers. Okay, so let's say we just blithely don't care about outliers. Yeah?
When you say adjustment, you sort of can
I'm going to have to come to you. I talk loudly so I can hear myself think. Let me-
So when you say adjustments, is it a form of linear regression?
Yes, it's just so the question was, when we do the adjustment, is it just linear regression? Yes, we could do just a simple linear regression and provide the user with the residuals, but then they'd freak out because a lot of the brain weights would be negative, and the molecular biologist would go, what's a minus brain weight? And then we'd have to explain residuals, so we'd simply compute the residuals and then add the constant. Is it? Yeah, okay. So so for those of you who are here, you could ask different questions. You could say, forget the genetics, I'm really interested in, is there a relationship between brain weight and some kind of behavioral trait, right? So then you'd say, calculate the correlations with everything else. And you'll notice that it gives you the choice of two types of correlation, and after this week, I'm really tempted to add some other measures here, which are robust, but maybe don't lose as much power as Spearman rank. Spearman rank with this sample size shouldn't be a problem. Pearson obviously will be sensitive to that outlier. But this is the advantage. Let me introduce you to a concept called Windsor Rising. It stands for the name of a statistician, but I always think of it as Henry VIII cutting off all of his wife's heads if they got annoying. So if you have an annoying data set or data trait and you want to clip it, you just clip it off. So in this case, I'm going to Windsorize the top two. So I'm basically making them, and I'm going to add just a little bit so I keep the rank the same. So I've basically trimmed the top of my distribution, and when you Windsorize, you're supposed to Windsorize symmetrically. I don't think it really matters, but this is what people think is the kosher way to do it. So I've trimmed the bottom and the top equally. I don't have any outliers now. And if I go back here and I do the basic statistics and update the figure, you'll see what you expect to see, which is my QQ plot now is truncated at the bottom and at the top. So I don't have undue, those points don't have undue effect anymore. So that's why we try not to database everything and lock you into the data. Because if you're doing this in a multivariate space, it's even more important, because you might have everything just look wonderful in univariate space, but be multivariate non-normal, and you have to basically figure out what to do in a situation like that. So that's, again, why everything is sort of live. Okay, so I have now cleaned up my trait, and now I want to map it. So we've looked at this before. Oh, let me just show you all of these basics. Okay, so QQ plot we saw. This is before I cleaned it up. That you saw, actually no, you saw this. This is the rank, data by rank, and you can see that BXD5 really is a super outlier, a very brainy mouse. And by the way, all of these, you can just sort of pull them off this page. They're GIFs. Unfortunately, they're not vector graphs. We're redoing GNetwork, so they're all vector. So you can put them into Illustrator or some other cool program. But right now, they're fairly high quality GIFs. Okay, so then we're going to map. So the mapping tools are down here. And there are 4 mapping tools right there. There's interval mapping, marker regression, composite, and pair scan. And they're progressively, well, they're slightly different models. So marker regression is what I actually showed you. You've got one marker and one trait, and you're asking, what's the t-test score? What's the, you could do it any number of ways. You're basically asking, what's the p-value of that relationship, marker by marker, and you don't care whether you've got a big gap or not. Interval mapping does the same thing as marker regression, but it estimates genotypes in intervals where you don't have an explicit marker. So it'll guesstimate, essentially. And that's the default position. Composite interval mapping is just partial correlation. It says, and we can do this in a second. It says, hey, I mapped my trait brain weight, and if you've done it already, you know that there's a QTL on chromosome 19. I know that I have something on 19. Do I have something else that's underneath that I might be able to fish out if I controlled for chromosome 19 and partialed that out? So when Steve was telling you about structural equation modeling, there's a baby step before there that if you don't know about it, you should learn it. Just go do a wiki search for partial correlation. You know, A covaries with B, A covaries for B, controlling for C, D, and E. So you're partialing out C, D, and E. For example, I'm interested in body weight versus age, but I want to factor out sex. So you could partial that out. And in this composite interval mapping, we just partial out one marker. And we're asking what kind of other SNPs or variants control brain weight. And then PairScan does an explicit test for epistatic interactions. So that was a question Anita asked earlier. You can do it in the BXDs because the sample size here is way big enough for mouse, but it would never be big enough for human. I could show you each one of these in turn. So let's do first marker regression, because this is what you'll typically do in a human GWAS study. There's no reason to impute. Aha, it says, Rob, you idiot, I don't want you to spend another $10,000, so this is a thing we put there. Do you want us to remove outliers for you? So let's say, okay, fine, let's remove the outliers. And now, basically, it did something a little hardcore here. Just threw away 2 of my high brain weight cases. It threw away BXD5 and BXD whatever. And here's what people call a Manhattan plot if it's done in a human GWAS study. We have the chromosomes, and then we have our chi-square statistic, the likelihood ratio statistic. Again, this is LOD1, LOD2, LOD3, LOD4. And in a human GWAS, you'd need LOD8 to get excited because there's so many multiple tests. Here, we're excited if we get up to about 16, or a LOD of 3. So that pink line is empirically determined by permutation. So every time I run the simulation, or every time I do the mapping, that pink line will bounce around a little bit. Not very much, because we have 5,000 permutations. Okay, and this is what's called the suggestive interval. So these are kind of sort of interesting spots, but the main one you can see is over on 19. But the question is, what about this one on chromosome 1? It's almost significant. If I controlled for chromosome 19, would it become more significant or less significant? So that's where composite interval mapping would be a useful thing to do. So let's, down here, it tells us which markers are the most important. So let me put that marker. Let me just remember what that marker is. It's got this silly name, UT whatever. So now I'll do the next thing. I'm just going to work through these in turn. So the next most complicated thing is simple interval mapping, and that's what it's caused. This time I'm going to take the outlier out to see if, or leave it in, I'm sorry, and see if that affects the value, and you could just kind of compare and contrast. It's not going to affect it much, as I recall. Okay, so there's my QTL on chromosome 19, slightly visually better. And let me explain some of the other colors here. So you guys know what the LRS is in blue, and that's the blue function that you see going through here. Well, what about these other colors? Those are the effect sizes. How big an effect does an allelic substitution going from a B to a D contribute to brain weight? And I'll show you where that is. So it's actually on the other side of the graph is the effect size, which is in whatever units you measured. So we're measuring brain weight in milligrams, that's in milligrams. So we have 5, 10, and 15 milligrams. It means that at this locus on chromosome 19, the red goes up to here. So there's about a 10 milligram effect for the addition of every B allele. So one of the parents is double D, right, DD on both chromosomes, because these are inbred strains. The other is BB. And if we swap out a D for a B, we gain 15 milligrams of brain weight. So that's called the effect size. And if you ever read a genetics paper and it says, we've discovered a QTL for intelligence or prostate cancer or whatever, you want to know, if you have the p-value, it doesn't really mean, I mean, it's nice to know that's real, but really what you want to know is what's the effect size. And the effect size will typically be measured in relative risk. So there's a small increase in your likelihood. I just mapped with the Enigma group, Paul Thompson and colleagues, a gene or SNP in humans that controls the size of the caudate nucleus, the putamen, part of the brain that's involved in Huntington's. The gene is connectin 1. It accounts for a difference in 2 milligrams of putamen weight, which is like about 1% or less. And that's a big effect. That was the biggest one we found, 20,000 people to get that one gene, which we then kind of validated in the BXDs. And here's a sort of sad story. So Lulu and I and 2 others were the mouse component of Enigma. It had about 400 authors. So the paper is that long. It's a short report in Nature. The author list is this long. And there are 4 of us mouse people in the middle of this. And when we sent it in with the mouse part, the reviewer said, we like the human part, but this mouse stuff is just crazy. It was 2 lines. And they made us take it out, but Paul graciously let us stay in there. But we showed that the gene in human also controls striatal, or is co-varies with striatal expression or striatal volume in mouse. Okay, enough of that. I don't know where it's going with that, but it's a fun story. Damn those reviewers. At least we got it in.
I would like to ask you something. So this is just for one mouse line?
This is for one mouse family. So we have a family here. I think the sample size here is about 80 members of the family.
So, and if I would like to order this mouse line, how can I get the information? Because at the beginning I don't remember if you choose a specific number, which you showed before, like you showed a lot with the different numbers of them.
Yeah, let's go back a little bit to answer that question of what did we do? How did we get here? So let me just review how we got here. So here are brain weights from all of these mice now organized from high to low or low to high, or I can just go by the index. So here they are originally. I've tinkered with it. Let me reset it. So now I'm reset. Okay. So you can see I filled in the outliers, BXD1 and 5 that I deleted.
So what is the index stand?
The index just is an index, just so you don't, yeah, that's just a dumb index. So if you export it, you know if you deleted one by mistake. The name of the kid, so this is kid 11 and kid 12 and kid 13, right? And so you could say, oh, I want to buy BXD5 or I want to buy BXD1, but in general you have to do the analysis across the whole set, right? So if you want to know, are mice with big brains smart, you just would answer that question just by saying, compute the correlations between all the other traits that have ever been measured. So that's percolating in the background now. But let me go back to mapping. So you've seen these 2 mapping, marker regression, interval mapping. Composite. I still have that marker in my clipboard. So that's the marker that I want to control for. Now I want to know, do I have a secondary QTL? So now I'm sort of getting to a network, or at least I'll have potentially 2 markers that control brain weight. So I'm going to compute that. I'm going to keep my outliers. And while that's computing, and this happens too quickly to really, too quickly. So you can see that by controlling for chromosome 19's effects and looking for secondary effects, my locus on chromosome 11 actually got smaller, but this locus on chromosome 11 got a lot bigger. And this is one we've actually mapped a long time ago. I've mapped that in many different crosses, or 2 or 3 different crosses. I know that one's real. as a locus, but I'd love to get the gene. So now I have 2 markers that I'm pretty sure control brain weight. How much brain weight does this one control? Well, the red is about there. So we could go over there and see how many milligrams we've got there. So it's almost another 10. So that means the difference between DD and BB, we've got 2 times 10 times 2. So we're accounting for quite a bit of the variance in brain weight here just with 2 markers, which is kind of nice. And now you can see where we're getting to a structural equation model, right? So now I can take markers in those 2 loci, and maybe I can substitute the markers with gene expression data in those QTLs. So I could take candidate genes, and I could use the variance in the candidate genes themselves instead of the markers to predict brain weight, and maybe, quote, clone some gene weight QTLs, which would be very cool. So that was the exercise I was asking you to do. How many people got sort of to the 19 on their own? Good for you. Okay, all right. Okay, so now it's exactly 5. Steve, guidance. What do we want to do? It's exactly 5 now. Yeah. Questions we can do for sure. How long?
Well, in principle, if you have more material, you could present it now, or we just...
I have all of tomorrow. When do we normally cut?
We end at 5.
Okay.
Officially.
Okay, so you guys should have a pretty good feel. You now understand mapping to some extent. What we haven't done is really network analysis, and that's what I want to do tomorrow. So tomorrow will be really bringing bigger batches of traits together, computing eigengenes. So Gene Network computes eigengenes beautifully, very rapidly. You get as many PCAs as you have subjects that go into the analysis. So, yeah, Anita.
So I don't know how much it does apply. So when you do a human GWAS study, you are dealing with the stratification. And the stratification problem is coming from the LD, when you are trying to put different population together. And you need to,
Correct for it.
Correct for that.
Right.
So my understanding when you are putting together all this, just tell me if I don't understand, putting together all this whatever line you have, you are dealing with different LD because you are making them having different LDs. So do I need to, or are you correcting for this one, or do you have this certification programming even?
Yeah. So to rephrase the question, if you have one family, you don't have problems of stratification. You have one mom and one dad. The BXD family, we don't have much of a problem. We have a little problem. So there is some stratification because the family was actually made in 3 cohorts. If you're dealing with human population and you have 20% African American, 15% Hispanic, 70%, I don't know if my math adds up here, the rest are Caucasian, what the heck do you do? Because you have very different allele combinations in the different populations, and we call that stratification. And the solution actually was developed here in the LA basin by a lot of people, but basically it involves using a general linear model, and you compute a kinship matrix. And a student of mine that worked with Eleazar Eskin and got his PhD here, a guy named Nick Furlot, took some code that was developed at Microsoft of all places called FastLMM and implemented that in Python, and it computes a kinship matrix. So basically you can throw the kitchen sink into the analysis, it computes the kinship matrix, corrects for the stratification, potential stratification artifacts. It's not implemented in Gene Network 1, but in Gene Network 2, which is our new cool thing, which I'm showing you here, it corrects for all of that, for any population you show at it. And for those of you, there's somebody from Abe Palmer's lab here. Where are you? So in your case, you've got this very complex relationship among inbred strains, and it'll correct for that if you give it the right genotype. So one strain of mouse is not the same as another strain of mouse. So they have a structure. And so if I were to do that here, So here's the human data set. Let me just show you the human data sets. Some of these are cool. Here's all of the GTEx data. This is the only place where you can map the GTEx data. So I don't know, I'll probably go to jail in the next few years. You read about me in Science, because I've made the GTEx genotypes available to users behind a firewall, so you can't actually see them, but you can use them. And the Bro doesn't do this, but let's say I look at sonic hedgehog in adrenal gland, and I've selected it. And now the mapping tools here include this Python linear mixed model, RQTL, Plink, which is the common one, and then GEMMA also corrects for kinship matrix. This is from Matt Stevens' group at University of Chicago. So in GNetwork 2, we're adding much more sophisticated mapping tools, but the interface isn't as visual yet, but it'll do all of that stuff. So yeah, play around with GNetwork. If you have specific questions or things you want me to do, I'm happy to kind of do homework for you or work with you tomorrow or even tonight. So or send me an email and say, I'm interested in this trait, where's the best data? Because one of the things I didn't tell you is, how do you find data that's relevant for your problem? Maybe you want high-fat, low-fat diet, or maybe you want a neurodegeneration data set that's young and old, like we heard about for Huntington's. We often, probably 50% of the time, will have something that's relevant for you in GNetwork. So be happy to get labwilliams.gmail or rwilliams.uthsc will get to me.
Thank you very much.